This is Chapter 24, Mini Lecture A. We're going to talk about metabolism and cellular respiration. So metabolism is the sum of all the chemical reactions happening in the body. Those that require energy, those that consume energy, which is the same thing, energy processes within the body. Many factors contribute to overall metabolism, including the lean muscle mass. The more muscle we have, the more metabolic requirements we have. The amount and quality of food consumed, and the physical demands that we place on the body. So all of this metabolism, our cellular respiration, is driving at creating ATP. ATP is our energy currency, right? It's our energy dollars. And that molecule is right here. That's our ATP. And this is showing what happens when we spend it, right? So when we spend our dollar, we spend our ATP dollar, we're going to put that energy someplace else. We're going to put that phosphate someplace else, and that's called phosphorylation. Phosphorylation, when we stick that phosphorus onto some other molecule in the cell. And as we do that, we split off one of these phosphates. Here we've got our ATP with our 3 phosphates. We split off one of these phosphates, stick it on something else. This is showing us one. It's not showing it stuck on, it's just showing it loose. And here's our ADP, D for di, adenosine diphosphate, right? So over here we're spending, and over here we're making, right? So over here we're adding that phosphate back onto the ATP, right? So this is our ATP, our energy currency. When we take that phosphate off and stick it someplace else, that's called phosphorylation. We're phosphorylating some other molecule in the cell. We're doing some kind of work in the cell. And then in cellular respiration, we are putting that phosphate, we're putting this phosphate back on. And so that's cellular respiration, which we'll talk about here in a second. So cellular respiration, what is cellular respiration? If we're alive, we're doing it. So in cellular respiration, we're going to use a sugar or carbohydrate. In this case, C6H12O6 is representing glucose or other carbohydrate, but glucose is a primary thing we want to use. Oxygen, O2, and this is happening in the mitochondria, and we're going to yield carbon dioxide, water, and ATP. This was a mistake here, ATP. So we're not creating energy. Remember, we cannot create or destroy energy. We can convert it from one form to another. In this case, we're taking that energy in the sugar molecule and putting it into the ATP. We can think of how the body systems all support this reaction. For example, with our sugar, this is going to be provided by the digestive system. So we're going to digest that starch, that plant starch, into glucose, absorb that into our body, and then use that to power this reaction. Oxygen we're getting from the environment by way of our respiratory system, and we're getting it to the cells using cardiovascular system. The carbon dioxide will be taken in a reverse direction. The blood will carry the carbon dioxide to the lungs where we can breathe it out. So we're exchanging that carbon dioxide for oxygen in our lungs. We're going to get a water. We're going to get an ATP. We are not creating energy. So don't say that the mitochondria is making energy. It's converting energy from sugar to ATP. So over here we also have an ADP, and we have a phosphate group that we're going to have to stick on there, right? And this is happening in the mitochondria. So here's another look at that. So the first part, we're kind of looking at the overall view of cellular respiration. That's a T. Outside of the mitochondria, we have a process called glycolysis, in which we can take a glucose molecule and switch it into 2-pyruvate, pyruvate or pyruvic acid. And in this process is an enzyme called phosphofructokinase, and it's sensitive to the levels of ATP in the cell. So if we have high levels of ATP, we're just not going to even proceed with this reaction, right? We're not even going to proceed with glycolysis. If the ATP levels are low, then the phosphofructokinase will become active, and it will allow this glycolysis process to proceed. This process has several steps in it and involves several enzymes, and is going to yield us 2 ATP. We have 2 in, and we get 4 out. So we have a net yield of 2 ATP. The pyruvic acid will then move into the mitochondria, be converted into acetyl-CoA, and then we have the citric acid cycle and the electron transport and oxidative phosphorylation chain. And this is where we get out most of our ATP. You see great big ATP. So we have basically a 3-step process in cellular respiration. This is the overall reaction, but we have glycolysis happening outside the mitochondria in the presence or absence of oxygen. The citric acid cycle and electron transport chain can only occur with oxygen. Oxidative phosphorylation. So oxygen is the final electron acceptor in this process. So this is why oxygen drives the whole process, right? Oxygen is driving this. There you see that oxidative phosphorylation. All right, catabolic versus anabolic. Catabolic metabolism is when things are broken down. So this would include digestion and is going to include hydrolysis or breaking with water. So in hydrolysis, we're breaking apart those macromolecules into their monomers, and we have to spend a water molecule to do that, right? So the hydrogen is stuck on one end of the monomer, the OH on the other end, and we call that hydrolysis. Anabolic, we're building those molecules back up, so now we have those monomers in the cells, and we're recreating the macromolecules that we need in that particular cell. And in that process, as we stick those 2 monomers together, we get an H off of one and an OH off of the other. That becomes a water, so we call that dehydration synthesis, right? So we're sticking the monomers together, getting water off. We call it dehydration synthesis. Catabolic is breaking down. Anabolic is building up. Reduction versus oxidation. Oxygen is going to steal an electron. So something that gives up an electron has been oxidized. Something that gives up an electron has been oxidized. Something that receives an electron has been reduced because an electron has a negative charge. So if we've added electrons, we've reduced it, right? We've reduced the charge. And so everything, pretty much everything's a redox reaction. All right, sources of ATP. So during catabolic reactions, proteins are broken down into amino acids, lipids are broken down into fatty acids, polysaccharides are also called starches, or glycogen are broken down into monosaccharides, and then those are transported to where we need them, and these building blocks are then used for the synthesis of molecules in anabolic reactions, right? So this is just showing us our food. We've got digestion, and then we carry these things to the parts of the body where we can use them. All right, so this is just a different illustration for cellular respiration. This is the part with the glycolysis happening outside the mitochondria. It can be with oxygen or without. We're starting with a glucose, and we're getting 2 pyruvates. In that process, we are contributing 2 ATPs to start, and we're contributing 2 NAD plus to start, and we're getting out 2 pyruvates, 4 ATPs, and 2 NADH. Okay, So we're getting these 2-pyruvate molecules, which we can use then either in the mitochondria, or we can do some other things with. This portion is all happening in the mitochondria. So this is just a different representation of those steps we talked about. So we turn those pyruvates into acetyl-CoA. We take that acetyl-CoA and we go to the Krebs cycle. I am not going to require you to memorize the steps of the Krebs cycle. We're going to look at it in a second. You could know that the carbon dioxide is coming off at this point. We're also getting carbon dioxide off at that point. And then this is representing the electron transport chain. And we'll see that again in a minute. This part is happening across that membrane inside the mitochondria. And this is where we're really pulling off our ATPs, is on the electron transport chain, with that oxygen just grabbing for those electrons. The oxygen just wants electrons, and it's just driving that part of the reaction. All right, this is our glycolysis overview. So this was that first little step. We're starting with glucose. We're ending with 2-pyruvates. We're putting in 2 ATPs. We're getting out 4 ATPs. And we're getting out 2 NADH. This is our rate controlling step, right, where our phosphofructokinase is in here. And if the ATP concentration in the cell is high, this will not go forward. If ATP is low, then yes, this will go forward, and we will allow this reaction to proceed so that we can make some more ATP. All right, aerobic versus anaerobic. Aerobic is with oxygen, anaerobic is without. Aerobic is with oxygen, anaerobic is without. Think of the exercise class called aerobics. That's specifically designed to keep everybody in the aerobic, not in the anaerobic, right? So anaerobic would be something like sprints, or maybe lifting weights until you get the burn in the muscle. But aerobic is deliberately, just aerobic exercise is designed to keep you over here in this aerobic portion, and that allows you to ultimately burn more calories because you can keep going longer. So this side is with oxygen, where we have our glycolysis, and then we proceed through the rest of cellular respiration in the mitochondria. In the absence of oxygen, then we go to lactic acid. And this is the burn, right? So if we're lifting weights and we get that burning feeling in the muscle, that's lactic acid. Generally, it's going to clear away pretty quick, right? As soon as you stop and allow the blood flow to return, you're going to clear out that lactic acid. And so that just occurs because the body can't get the oxygen there fast enough, right? If you're doing a sprint, or if you're really lifting weights really hard and fast, your blood supply just can't get the oxygen there quickly enough. So once you stop and allow the blood to return, the lactate will clear out. All right, the Krebs cycle. So this was that stage in the mitochondria. You do not need to memorize all of this. Know that it is a cycle. There are many enzymes involved. And then we have these intermediate molecules, and some of those can be used and pulled off into other pathways. Right. So we're using those 2 acetyl-CoAs in this cycle and going around, and we're producing NADH, FADH2, and ATP. So we're making these high energy molecules in this cycle. All right, electron transport chain. So this is the stage. This is happening across that inner membrane of the mitochondria, and we're getting these sodiums moved across, and they are going to come back through this enzyme called ATP synthase. ATP synthase. So anytime we have that ACE on the end, we know it's an enzyme. And when we have synthase, we know that that's a making, right? Synthesis is making. Sorry, that didn't fit. It looks like a murder scene on there. All right, so this is the ATP synthase right here, and it actually rotates. As this sodium comes back through, this giant protein rotates and makes ATP. So it's phosphorylating that ADP into ATP. It's adding this phosphorus on and making ATP, right? And releasing a hydrogen ion at the same time. So this is a series of electron carriers and ion pumps to get that hydrogen across, right? And now we've got our ATP as that comes through. There's some nice animations on this online, and this thing rotates. So that's ATP synthase is in the membrane, in that inner membrane of the mitochondria. All right. So this is, again, just linking those all together, looking at what happens. So there's our glycolysis, there's our acetyl-CoA, there's our Krebs cycle, electron transport chain, and there is our net ATPs, right? We're putting 2 ATPs in, we're getting 4 out, so that was a net of 2. Can't really read this. Look in your book and go over this carbohydrate. Don't spend a whole lot of time on Krebs cycle. Read through it, but don't let it get too bogged down. All right, gluconeogenesis is the synthesis of glucose from pyruvate, lactate, glycerol, alanine, glutamine. These are amino acids. Alanine and glutamine are amino acids. So we talked about that pyruvate and that lactic acid. So we can use that lactic acid, right? So let's say we did that. We lifted weights until we had the burn in the muscle. We can then later use that lactic acid to make glucose. So let's look at this word, gluconeogenesis. That's making glucose new. So neo is new, genesis is making and glucose, right? So we're making glucose new. This is going to happen in the liver. So there's our glucose, whole long process. So we're using up these different little metabolites. All right, triglycerides or fats, right? So there's our 3 fatty acids on the triglyceride. In this case, we've got a different chain length for each fatty acid. This is one of our energy storage molecules. This is our long-term energy storage, right? And we're going to get into this when we have used up our glucose and our glycogen, right? So we can use up glucose pretty fast. Let's say we're going on a hike, then we're going to use up our glycogen, and then say we keep hiking and we're not eating anything, then we're going to get into our triglycerides, right? So there's our 3 fatty acids and our glyceride molecule. All right. Chylomicrons contain triglycerides, cholesterol molecules, and other apolipoproteins. So that's going to be, lipoproteins are going to be molecules that have both protein and lipid as part of the protein. And notice that we've got a phospholipid around here. So this is going to allow us to carry lipids around the body, right? So remember, lipids are not going to be soluble in water. So these function to carry those water-insoluble molecules from the digestive system through the lymphatic system and into the bloodstream. And there, those lipids can be carried to the adipose tissue for storage. Right? So our purpose here is just to carry these through the blood system because they're not water soluble. Right? So we've got all those phosphate heads that can be soluble in water. Breakdown of fatty acids. All right, so this can give us acetyl-CoA. So this can be used for energy when glucose levels are low. We can also use proteins for, right, when they're present. All right, then if we have excess acetyl-CoA, can be diverted from the Krebs cycle to the ketogenesis pathway. And this can occur in the mitochondria of the liver cells. And this results in beta-hydroxybutyrate, which is a ketone body found in the blood. All right, so this is just another metabolic pathway happens in the liver. There are about 500 functions in the liver. Not exaggerating there, hundreds of functions. All right, when glucose is limited, we can oxidize those ketone bodies to produce acetyl-CoA to be used in the Krebs cycle and generate energy. All right, and lipids can be used in metabolism as well. And then let's look just for a second at digestive enzymes and hormones. We're going to come back to digestive system later. So we have an enzyme called amylase produced in the saliva, and that's going to break down starch. So we're going to start the breakdown of starch in the mouth. Then we're going to start the breakdown of protein in the stomach. So the stomach, we have proteases and we have hydrochloric acid. And that's all we're digesting in the stomach. So that amylase up here is actually denatured when it gets to the stomach. And then just as we leave the stomach, right about in there, we have this little gadget. This is the pancreas. We're not seeing it too well. It's behind the stomach. And the pancreas is producing a number of digestive enzymes. Enzymes are going to break down protein, lipids, carbohydrates, nucleic acids. We also have sodium bicarbonate, which is going to neutralize the hydrochloric acid coming out of the stomach. And all of that is dumped into the small intestine right there. We also have some bile coming from the liver dumped in. That's going to help us break down the lipids so that we can digest them more easily. And then all here through the small intestine, we're going to be digesting those macromolecules and absorbing the monomers. All right, that is the end of, not quite the end. What else do we have here? We talked about hydrolysis. Phosphorylation is adding a phosphate and adding energy at the same time. We talked about reduction and oxidation. We talked about hydrolysis and dehydration synthesis. We talked about ATP synthase. We talked about oxygen as the final electron acceptor. Breaking down molecules is catabolism. Building that back up is anabolism. All right. That's the end of the lecture, mini lecture.